A miniature horse can stand barely taller than a large dog, and a Shire, the very same species, can stand taller than a grown man. It's easy to assume that gap means centuries of separate development, maybe even different species. It's not just size. Some breeds move in a smooth, gliding gait that most horses can't perform at all. Some are covered in spotted patterns so bold they look almost hand-painted. And some carry a hidden, testable health risk tied to the very trait that makes them famous. Most owners have wondered at some point why their horse looks nothing like the one boarded 3 stalls down. Some are famously hot-tempered. Others stay unshakable around chainsaws and city traffic. Once you know what's actually behind it, a paddock full of totally different-looking horses stops looking random and starts looking like the same animal, rearranged. This is the genetics behind one horse, 300 breeds. It starts in one place, one small population of horses. Everything else follows from there. That ancestor lived about 4,200 years ago, on the grasslands of what's now southern Russia. From there, breeders mostly didn't invent new biology. They found what already existed and pushed it further. A handful of genes shape size. One single gene decides gait. A few more shape color and pattern. One of those patterns is older than domestication itself, and it's painted on the walls of Ice Age caves. Coming up, the same species stretched further than almost any other domestic animal alive. Start with the most visible difference of all, size. A Falabella can stand under 76 centimeters at the shoulder, about 30 inches. A Shire or Percheron can clear 2 meters, about 6'6" at that same point. That's roughly a 2.5 times difference in height, inside a single interbreeding species. Researchers scanning the genome across small and giant breeds found the pattern wasn't scattered across hundreds of genes. It was concentrated in a small number of specific regions on the horse genome. One region, called LCORL and NCAPG, stood out. Its alleles almost perfectly split large breeds from small ones. 2 more regions, HMGA2 and ZFAT, layer on top of that, fine-tuning the range further. Together, this small set of gene regions accounts for most of the size difference across horse breeds. Miniature and Shetland-type ponies carry an extra layer on top, 4 more variants that specifically suppress growth. All 4 sit in the same growth pathway, working almost like an interlocking brake system on the body. Ponies carrying all 4 variants never grew taller than about 87 centimeters at the withers. That specific set of variants looks to be genetically recent, likely only 12 to 1,800 years old. Not every breed sits at an extreme. The Arabian keeps a build closer to where this story started. Size was the clearest case. Gait turns out to be even more precise. A single gene, a single switch. Some horses can do more than just walk, trot, and canter. Icelandic horses can tolt. A smooth 4-beat gait a rider can sit through without bouncing. Standardbreds, bred for harness racing, can pace, both legs on one side moving together. Most horses can't learn either gait, no matter how skilled the trainer is. The difference comes down to one gene, DMRT3. Researchers informally call it the gatekeeper. DMRT3 is active in the spinal cord, in circuits that pattern how a horse's legs coordinate at each step. One version keeps a horse to the ordinary 3 gaits. A single mutation opens the door to the rest. Quick note, if you've seen our video on why horses are rideable at all, that's a different gene. This one decides how a horse moves, not whether it can carry a rider. Ancient DNA traces this mutation to a specific place and time. Medieval England, roughly 850 to 900 AD. From there, it likely traveled with Viking-era horse trade north to Iceland. Icelandic breeders then selected it hard, generation after generation, until it became the population's default. By 2012, close to 94% of Icelandic horses carried 2 copies of the mutation. The same gene shows up an ocean away in standardbred harness racing. Same mutation, a completely different context. One mutation, 2 very different horse cultures, centuries and continents apart, both selecting for the same trait. Color and pattern turn out to be one of the most tightly mapped parts of this whole story. Almost every classic base color, chestnut, black, and bay, comes down to just 2 genes working together. One gene, MC1R, can switch off the signal for black pigment, producing chestnut. A second gene, ASIP, decides whether black shows only at the points, giving bay, or the whole body. White spotting is a different story, the same chromosome as the size genes, but a completely unrelated gene called Gait. One specific rearrangement in that region produces tobiano, bold, rounded white patches that cross clean over a horse's back. Related variants in that same gene region produce sabino, roan, and dominant white. Several distinct looks, one shared neighborhood. Then there's the pattern that looks the most invented of all, and turns out to be the oldest. Leopard complex spotting, the Appaloosa look, comes from a single insertion inside a gene called TRPM1. Depending on how many copies a horse carries, the pattern ranges from a light blanket to a full leopard coat. Here's the part that changes the whole picture. This pattern isn't a breeder's invention. Researchers sequenced coat color genes from genuinely wild pre-domestication horses that lived during the Ice Age in Pleistocene Europe. 4 out of 10 of those wild horses already carried the leopard complex genotype. It matches something else, too. Spotted horses painted on cave walls, tens of thousands of years before domestication began. Humans didn't invent this pattern. They found it already there, at low frequency, and bred it up. That same mutation carries a second effect, one that has nothing to do with looks. Horses with 2 copies also have congenital stationary night blindness, measurably impaired vision after dark. One coat pattern, one ancient origin, one linked cost that never makes it into a listicle. Zoom out, and the same pattern keeps showing up across all of this. Size traced to 4 gene regions, gait traced to 1, DMRT3, color traced to 4 more, including TRPM1. And the timeline behind all of it is short. The first formal horse stud book wasn't opened until 1791. Most breed registries alive today are only a century or 2 old. That's intense human-directed selection, compressed into a genuinely short window, not gradual evolution spread across the whole genome. Breeds with old, closed stud books show measurably less genetic diversity within the breed than newer, more open ones. That's exactly what you'd expect if closing a population and selecting hard is what compresses it toward an extreme. Study after study, largely the same genes keep reappearing, not a different set each time. The most extreme versions we see now, the tiniest minis, the most saturated leopard coats, came from deliberate breeding. Most of that intensity landed within just the last 100 to 300 years. A short genetic window, a small number of genes. That's the entire mechanism behind 300 breeds. This isn't just spectacle. Some of it is genuinely useful if you own one of these breeds. If you own or are buying a leopard complex Appaloosa, the LP gene can be tested for directly. Horses with 2 copies, homozygous for it, often have that congenital night blindness we mentioned. In practice, that can look like extra anxiety or spookiness specifically after dark, not a training problem. It's worth ruling out before assuming it's a behavior issue. Miniature horses bred hard for smallness carry their own consideration, an elevated risk of skeletal dysplasia in some lines. If you're breeding minis specifically, that's worth screening for, not just selecting for size alone. A few honest caveats, so this doesn't oversell itself. 300 breeds is the commonly cited figure, but real estimates range from roughly 250 to over 600. Not every breed trait fits this fast human-driven story either. Small, hardy island and mountain pony types likely faced genuine, slower environmental pressure over centuries, alongside selection. What's fast and deliberate is the extreme end. The smallest minis, the biggest drafts, the most saturated patterns. None of this makes these traits purely cosmetic either. Several carry a real linked physiological cost, not only a look. So the next time you see wildly different-looking horses side by side, remember what you're actually looking at. You're not looking at 300 separate evolutionary experiments. You're looking at one species, one small toolkit of genes, refined over 4,200 years. Nature supplied the raw variation. We supplied the intensity and did it astonishingly fast. I used to see a Shetland pony and a Shire as almost unrelated animals. Now I see the same 4,000-year story, just pushed to opposite ends. If this changed how you look at horse breeds, subscribe for more science like it. New videos on the evolutionary science behind horse behavior every week.